As we age, our immune system undergoes significant changes, impacting our ability to fight off infections. One of the key players in this process is the CD8 T cell, a type of white blood cell crucial for controlling viral infections. Scientists have long observed that older adults are more susceptible to both new and recurring infections, but the underlying reasons are complex. Recent research has begun to unravel how both aging and chronic viral infections, such as cytomegalovirus CMV, jointly influence the diversity and effectiveness of our T-cell repertoire. In this video, we'll explore how age and CMV infection shape the immune response, focusing on the diversity of T-cell receptors and what this means for our health as we grow older. CD8 T cells are specialized immune cells that play a central role in identifying and eliminating cells infected by viruses. They do this by recognizing small fragments of viral proteins, called epitopes, presented on the surface of infected cells. The ability of CD8 T cells to recognize a wide variety of viral threats depends on the diversity of their T-cell receptors, TCRs. Each TCR is unique, generated through a random recombination process, allowing the immune system to respond to countless different pathogens. However, as we age, both the number and diversity of these T cells can decline, potentially weakening our immune defenses. T-cell receptor (TCR) diversity is a cornerstone of adaptive immunity. TCRs are generated through a process called VDJ recombination, which shuffles gene segments to create a vast array of unique receptors. This diversity ensures that the immune system can recognize and respond to a wide range of pathogens. The more diverse the TCR repertoire, the better the immune system can protect against infections. However, studies have shown that TCR diversity tends to decrease with age, particularly within the pool of naive T cells, which are essential for responding to new infections. With advancing age, the immune system undergoes several changes. The number of naive T cells declines and the diversity within this pool also decreases. This means that older adults may have a reduced ability to respond to new infections. Additionally, memory T cells, those that remember past infections, may become less effective over time. These changes contribute to the increased vulnerability of older individuals to infectious diseases and may also affect how well vaccines work in this population. Cytomegalovirus (CMV) is a common virus that establishes lifelong infection in most people. While often asymptomatic, CMV has a profound impact on the immune system, especially in older adults. CMV infection leads to the expansion of CMV-specific T cells, which can occupy a significant portion of the total T cell pool. This expansion may come at the expense of T cells specific for other viruses, potentially reducing the overall diversity of the T cell repertoire and affecting immune responses to other infections. Epstein-Barr virus, EBV, and CMV are both herpes viruses that persist in the body for life. The immune system is constantly challenged by these viruses, leading to the presence of high frequencies of virus-specific CD8 T cells in most individuals. Over time, repeated stimulation by these persistent viruses can drive changes in the T cell pool, including increased numbers of terminally differentiated cells and shifts in T cell phenotypes. Understanding how these viruses interact with the immune system is key to unraveling the complexities of immune aging. To investigate the effects of age and CMV infection on T-cell diversity, researchers studied healthy individuals across a broad age range. Participants included both young adults and older adults, with careful measurement of their CMV and EBV status. Blood samples were collected and analyzed for the presence and diversity of virus-specific CD8 T cells. By comparing these groups, the study aimed to disentangle the separate and combined effects of aging and CMV infection on the immune system. The diversity of the T-cell repertoire was assessed using advanced sequencing techniques. Researchers isolated virus-specific CD8 T cells from blood samples and sequenced their T-cell receptors. By analyzing the number and variety of unique TCR sequences, they could measure the richness and evenness of the T-cell repertoire. These metrics provide insight into how well the immune system can recognize and respond to different viral threats. The study found that both CMV-specific and EBV-specific T-cell repertoires decline in diversity with age. This reduction was observed regardless of CMV infection status, suggesting that aging alone is a major factor in diminishing T-cell diversity. The loss of diversity may limit the immune system's ability to respond to new or recurring infections, highlighting the importance of maintaining immune health throughout life. Beyond the effects of aging, CMV infection further decreases the diversity of the EBV-specific T-cell repertoire. Individuals who were CMV positive had lower diversity in their EBV-specific T cells compared to those who were CMV negative. This suggests that CMV infection can have a broader impact on the immune system, potentially affecting responses to other viruses beyond CMV itself. As people age and with chronic CMV infection, there is a noticeable shift in the phenotype of virus-specific T cells. Markers associated with terminal differentiation, such as KLRG1 and CD57, become more prominent. These changes indicate that the T cells are more differentiated and may have reduced capacity for proliferation and function. Such phenotypic shifts could contribute to the overall decline in immune responsiveness seen in older adults. One striking observation is the presence of large clonal expansions within the T cell pool, especially in CMV-infected individuals. These expansions consist of many T cells with identical TCRs, often specific for CMV. While this can provide strong protection against CMV, it may come at the cost of reducing the diversity available for other pathogens. This phenomenon, sometimes called memory attrition, is a key aspect of immune aging. To quantify T cell diversity, researchers use measures like richness, the number of unique TCR clonotypes, and Simpson's diversity index, which accounts for both richness and evenness. These metrics help distinguish between a repertoire dominated by a few large clones and one that is more evenly distributed. The study found that EBV-specific T cells had higher richness and diversity than CMV-specific T cells, but both decreased with age and CMV infection. To further analyze T cell phenotypes, the study used TSNE clustering, a technique that groups cells based on the expression of various markers. This approach revealed distinct clusters of T cells ranging from central memory to terminally differentiated cells. With age and CMV infection, there was a clear shift toward more differentiated phenotypes, especially among CMV-specific T cells. These findings underscore the dynamic nature of the immune system in response to persistent viral challenges. A common hypothesis is that the expansion of CMV-specific T cells leads to memory attrition, where other virus-specific T cells are lost due to competition for space and resources. However, the study found that while CMV+ individuals had lower frequencies and diversity of EBV-specific T cells, there was not a clear negative correlation between the frequencies of CMV and EBV-specific T cells. This suggests that memory attrition may not fully explain the observed changes, and other factors could be at play. The findings have important implications for vaccination strategies, especially in older adults. Since T cell diversity declines with age and CMV infection, vaccines may be less effective in this population. This raises questions about the optimal timing of vaccination. Should we vaccinate earlier in life to build a robust memory pool, or later to ensure protection persists into old age? Understanding the dynamics of T cell diversity is crucial for designing effective vaccines for the aging population. Most immune studies, including this one, analyze T cells from blood samples. While blood contains only a small fraction of the body's total T cells, it is often the most accessible compartment for research. For persistent viruses like CMV and EBV, blood may actually be the most informative site, as many virus-specific T cells circulate there. However, it remains to be seen whether findings in blood fully reflect what happens in other tissues, such as lymph nodes or the spleen. While this research provides valuable insights, there are limitations to consider. The study focused on specific viral epitopes and used cross-sectional data, which may not capture the full complexity of immune aging. Longitudinal studies tracking individuals over time would help clarify how T cell diversity evolves. Additionally, exploring other viruses and immune compartments could deepen our understanding of how aging and chronic infections shape immunity. In summary, both aging and chronic CMV infection contribute to a decline in the diversity of virus-specific CD8+ T cells, particularly those targeting EBV. This reduction in diversity may compromise the immune system's ability to respond to new infections and could influence vaccine effectiveness in older adults. The study highlights the need for tailored vaccination strategies and further research into the mechanisms driving immune aging. As our population ages, understanding how to maintain immune health becomes increasingly important. Strategies to preserve T cell diversity, prevent chronic infections, and optimize vaccination could help protect older adults from infectious diseases. Ongoing research will continue to shed light on the best ways to support healthy aging and robust immune defenses. The interplay between aging, chronic viral infections, and immune diversity is a complex but vital area of research. By unraveling these connections, scientists can develop better interventions to keep our immune system strong throughout life. Whether through improved vaccines, targeted therapies, or lifestyle interventions, the goal is to ensure that everyone can enjoy healthier, more resilient aging.